Hi everybody, my name is Dion Raskina and today I will be talking about G-protein coupled receptors and a couple ways in which they work. Before we begin, let me introduce to you Cadbury, my dog. He will be making a few guest appearances throughout this video and I hope you will enjoy seeing his cute little face. So without further ado, let's start talking about G-protein coupled receptors. G-protein coupled receptors, or GPCRs, are complicated and intricate and function to allow for a cellular response by an external messenger. The receptor is unique in that it has 7 membrane-spanning helices and is coupled with the G-protein in the intracellular portion of the plasma membrane. The G-protein has 3 subunits, the alpha, beta, and gamma subunits. The alpha and gamma subunits attach to the plasma membrane. In its inactive form, the alpha subunit of the G-protein is attached to GDP. An extracellular messenger, also known as a ligand, is the first messenger in the system and is responsible for signaling a certain cellular response. However, since the ligand is hydrophilic, it cannot cross the hydrophobic portion of the plasma membrane, so the ligand must bind to GPCRs in order to cause the desired cellular response. Let's say that in this scenario that the extracellular messenger is epinephrine and the target cell is in the liver. When epinephrine attaches to GPCR, the protein receptor undergoes a conformational change which signals the G-protein to activate. The alpha subunit is now attached to GTP instead of GDP and dissociates from the beta and gamma subunits. The dissociated alpha subunit then binds to adenylate cyclase, which is the effector protein. This activates adenylate cyclase, allowing it to convert ATP to cyclic AMP. Cyclic AMP is the second messenger and binds to protein kinase A. Protein kinase A is made up of 2 regulatory regions and 2 catalytic regions. As long as the regulatory regions of the protein kinase A molecule are bound to the catalytic regions, protein kinase A will remain inactive. When cyclic AMP binds to the regulatory regions of protein kinase A, the catalytic portions are released and activated through phosphorylation with ATP. The catalytic regions then bind to phosphorylase and activate the enzyme. Phosphorylase causes the breakdown of glycogen and releases the glucose molecules into the bloodstream, which in this scenario is the desired cellular response. The interaction of medicinal drugs and GPCRs can be seen in xylazine, which is a sedative and analgesic drug commonly used in veterinary medicine for the sedation of animals. Xylazine is an alpha-2 adrenergic receptor agonist. The alpha-2 adrenergic receptor is a type of GPCR. Veterinarians can couple the use of xylazine with ketamine in order to reduce the dosage of ketamine necessary for anesthesia during procedures such as surgeries. Xylazine is one of many important drugs that act on GPCRs. In fact, it has been estimated that about 30 to 50% of medicinal drugs act on GPCRs, which clearly indicates the importance of these receptors in the body. If the alpha subunit is an alpha Q subunit instead of an alpha S subunit from the previous example, then the first difference the pathway will exhibit will be when it binds to a different effector protein, phospholipase C. The activation of phospholipase C causes PIP3 to cleave into IP3 and DAG, which are secondary messengers in this system. IP3 binds to the receptors on the endoplasmic reticulum of the cell, causing a release of calcium into the cytosol of the cell. DAG activates protein kinase C, which can phosphorylate other proteins in the cell. Together, these cause the desired cellular response of the extracellular messenger. Great job learning about G-protein coupled receptors with me today. I think this calls for a tasty treat. I hope that this video helped you understand a little bit more about GPCRs and their various uses in the body. It is important to understand the function of GPCRs in clinical pharmacology or any veterinary practice. There are many more types of GPCRs that were not covered in today's video. However, the 2 that were covered are commonly found in different cells throughout an animal's body. Thank you for joining me today in learning about GPCRs. I hope you enjoyed it.